And I think you can see the complexity. David is, of course, being modest. We have about 20 people in the team at Leeds running haematology trials at David Leeds, with Catherine, our data side. And it's very efficient. We have to be very efficient because we have to report all the side effects. We have about 80 trials, I think, open at any point for haematology, across all leukemias and all. So we think everyone should have access to trials. So there are two, sort of, before I show you some slides about CLL trials, I'm going to talk about the CLL trials in the UK.
These are some questions.
That's some questions, okay. They write better than doctors, I can tell you. Oh, the first one does. So the two elements are, first of all, there's two, we've seen the phases of trials and they're very important, and the speed, you can see how slow it can be to get through a trial process, and that's really important because if we have an effective therapy, we want everybody to benefit from it quickly, so we have to get these trials done quickly. And what part of that's accelerating the process. So go from phase 1 to phase 3 can take 10 or 13 years. I mean, it can take a long time, historically, to recruit all the patients, to do all that safety work. And there's lots of blind alleys because sometimes the drugs don't work very well and you don't get to the end. So you have to pick the right combinations, for example, to test in trials. So we need to make that quicker, and I'll show you how we're doing that. And the second element to this is that there are two main types, sponsors. One sponsor is companies. So we work very closely with pharma companies, and two of those trials, at least, that David showed you, were sponsored by pharma companies. Because if we didn't have pharma companies, we wouldn't have any drugs. And we know that if you look at places where there's no pharma input, there's no drugs developed. And so we have to work closely with them, and they have to work closely with us. And we do that. We interact very closely with them. So some trials are sponsored by pharma to develop drugs. And the reason for the development of those drugs is to get a drug license so it can be sold. Now, my experience of biotech and pharma is actually that most of them are there for good reasons, not just to make money. They're there to develop drugs to do good for patients, and we need them. And so some trials are designed specifically to show a drug should be approved and sold. And then the collaborative groups, which I chair the UK group, and there's probably 3 major groups, or maybe 3 or 4 major groups in the world, will run trials that work with pharma and with the national groups to look at what treatment is best. Not just should a drug be approved, but what about combinations? What maybe we want to combine 2 drugs, one from one company, one from another. So we have to do those trials, and actually that evidence is really important going forward. Okay, so we have to embrace all of the phase 1 to phase 3. So I'm going to show you. Having said I don't do slides when I'm in front of the patient groups, I thought I'd show you some slides just to show that I never trust me. Okay. So the UK CLL has been very organised in terms of CLL trials for at least for many years, actually, for several decades, but increasingly so. And it's really important we accelerate the development of drugs for CLL, because until 10 years ago, there were really 2 or 3 drugs that were developed over a period of 20 years. And so we could take 10 years to look at a drug because another one wasn't coming. We've had about 10 drugs in the last probably 5 years. And so we can't be that slow. We have to be quicker in moving these forward so all the patients benefit, not just the 500 or half the 500 in a trial. And so one of our challenges is to do that. So what we're trying to aim, I'll just do this quickly, is we have, as David showed you, a mixture of different patients. Some patients are the average patient is about 70 when they're diagnosed. I've seen 3 teenagers with CLL. We see patients who are 93. We see people who are ill. We see people who have other side effects, other issues like heart problems. Some patients, the aim of treatment may be just to control these. If you're 88 and have a lot of comorbidities, actually just being controlled with few side effects is perfect. If you're 38 and you don't want to, 10 years isn't long enough, you need to be cured. And so we have to tailor it to the individual patients, first of all. There are different types of patients. There's mutated patients who are good risk and may not actually die of CLL, and then there's other patients with more aggressive disease. So we have to think about those patients, what's going to happen to the patients. The reason for not doing a stage A trial is some patients aren't going to need treatment, so why would we treat them? So we don't want to expose them to risk when we don't need to, but we definitely want to treat people who need to be treated. And then we've got frontline and relapse, and I'll talk a bit about that later on. We've got to make sure our treatments are tolerated well, and we have to report side effects. So not just as we're developing individual drugs, but in combinations, and that's really critical that we report the side effects, and we report everything. So one of the scary things of any drug you see, any trial, is the long list of side effects you see, and the pages and pages of that my arm could fall off, and could get hit by a bus, and all these sorts of things. Most of those things are nothing to do with the drug; they're to do with the disease or something else unrelated to that. But we report everything, because if we don't report everything, we bias it, and we may miss something important. So what you see is what's been reported in the trials, and we look at, particularly in those comparative trials, is one side effect greater for one drug than another, and then that tells us that it's due to the drug. And so that's why one of the reasons why phase 3 is very important, where we get randomized. Just for those who don't mean it means basically a computer decides. So the patients go in, we toss a coin, or the computer does, and decides which arm of the trial the patient's going to go in, and you stick on that arm. And we only do trials, and I think most centers only do trials if we're happy that both arms are appropriate treatments. So we wouldn't do it if we felt one was, and we have occasionally rejected trials because the standard arm isn't good enough, and so we've got better treatments. So tolerability, and then we have to think about the bottom, what we're trying to achieve. We're trying to cure the disease. So what we've done very effectively, I think, in the last few years is try to move these trials forward quickly. So what we used to do, for example, with the phase 3 trial, is probably discuss with people, companies, CRI UK, Bloodwise, for 3 years to get a trial open for a big phase 3 trial. And sometimes we went through blind alleys because we were competing with other international groups. Eventually, we get a trial open. We'd open it in 100+ sites in the UK. It takes a year, 2 years to open all those sites because it's painfully slow, I can tell you. And then it takes 4 years to recruit the patients. And then because we've got effective therapies, it takes 4 years before we prove it's better. So the whole process is much too long. So we have to shorten it. And so what we're doing is this. So we basically have a series of, as David explained, phase 1, phase 2 trials to really look at combinations. Does it look like it works? What's the effectiveness? We're using MRD as an endpoint, just so that shows us. I mean, it doesn't matter to somebody if they're MRD negative or positive. It doesn't make you feel better if you've got a little bit of disease or not, but it proves whether that combination is better than another combination. So it helps us move things forward. And then when we have that evidence, to rapidly move into the phase 3 trial, because that's what changes practice. We don't change practice on clarity or on those sorts of phase 2 trials. We change it on phase 3, and we have to do them quickly. Otherwise, a small number of patients will benefit in the trial, and the thousands of patients outside trials won't. So we have to get through phase 3 trials quickly. And we have to biobank samples. We have to store samples down from every patient in every trial at various time points, so we can go back and look at the samples and say, what's different about the patients who responded, didn't respond? Why did they respond well? Why didn't they respond well? What led to the resistance? So it's critical. And we've done this really successfully with the biobank in Liverpool. That's for the whole UK for CLL. And we've got thousands of patients stored down. It's really helpful because if we then develop another test, we can go back to all those cells that we've got stored and test it on our old trials. So biobanking is really critical. So if you're asked about biobanking in a trial, it's really, really important for us to move things quickly. So we've got, Bloodwise have been really great in terms of the trials that supporting early phase trials, and that's the charity we used to call the Leukemia Research Fund, or LLR. And we did a series of trials. So Icicle was a phase 2 trial. We treated 40 patients with ibrutinib. The reason I put this slide up, I'm not going to show you the data, is that these patients, as David said, have to come more often and have lots of more investigations, bone marrow tests, blood tests, scans, lots of much more intense than we would do in a phase 3. And here are the time points that we store samples down and we test it. Now, why it's important is that we're doing around the country, this isn't just in Leeds, but we've got several research groups around the country doing really impressive work. So we're doing lots of phenotyping, seeing all the different types of CLL and whether they respond, looking at how they respond to treatment, that's the same thing, looking at the genes and how they're switched on and off and how the treatment affects it, looking at resistance and looking at the genetics. So all this data is done on these 40 patients. And so we can really intensely study what's going on and learn how to do the next trial better. What combination, where should we be going to? So that's just an example of what we're doing in all of the phase 1, 2 trials. And then Clarity, which we've talked a bit about, and well, at least one person said they're on Clarity 2, is really our acceleration into combinations of two different companies' drugs, or they were originally when we did the trial. Now one bought the other, actually, which is interesting. So we're combining ibrutinib with venetoclax, and what we want to do is get these studies done really quickly, and that's what we've done. So this trial recruited in half the time we wanted, or just over half the time we wanted to recruit 50 patients. And our endpoint was depth of remission. So can we get negative remissions? And these were all patients who had had previous treatment, so they weren't frontline patients. And what we did was do this. So we worked out what we thought was safe and what was sensible, gave ibrutinib, added venetoclax, used a combination. And then the marrows and CT scans, if they're negative at 6 months, they stop at 12 months. So we're stopping treatment now rather than continuing. Or if they're positive at 12 months and negative at so if they're positive at 6 months and negative at 12 months, they stop at 24 months. So what we're doing now, actually, to make it even more rapid, is adding, hopefully, I'm negotiating, I spend my life negotiating, to add a drug here for the ones who are positive. So can we drive these patients to negativity by adding a third drug? So we can then learn more about how we use these therapies moving forward. I remind you, these are people who've largely failed chemotherapy treatment. And just to show one slide of results. So this is the depth of remission in the blood. So this is months, weeks and months of the trial. So month 8 is 6 months of the treatment. And you can see the percentage of disease. So at the start, in the blood, most patients have got 90% CLL. When we start adding venetoclax, you see some patients who are negative. This is 1 in 100,000 cells, the light green, and then the dark green is one more. And then as we go on, you can see it's improving with patients. So more patients are becoming negative. And our aim was to get 30% negative at 12 months. And at 6 months, we're already there. So that's a positive result. And that allows us to go forward. And the marrows, and I've already showed you the 8 month, it's the same pattern, and we'll get in marrows. So that's why the analysis is really important, because we really learn about how these drugs work in small numbers of patients. And then the really sort of important thing is moving to phase 3. So we have a FLIR trial. I mean, some of you might have been exposed to the FLIR trial, which is a frontline trial. So this is untreated patients at the top, 754 patients initially, to compare ibrutinib rituximab versus chemotherapy. So can we get rid of chemotherapy? And so that trial started in 2014. It's led from Leeds. In fact, I've got to rush off to do a trial steering committee meeting on it this afternoon, because it's independently monitored by other experts, and then that will stop effectively soon, because it will have 754 patients recruited. However, at the same time, we're doing these trials. I showed you Icicle in patients who have failed treatment, and so we're rapidly adding antibodies to it. I didn't show you that. Clarity, and showing it's effective. And as soon as we see a signal that says this looks better, what we've done is, which is, I think, new, really, is to change this trial. It's corrupted it a bit, and add 2 more arms to the trial. So at the moment, there are 4 arms open, because we want to know if this combination, ibrutinib and venetoclax, is better. And so we've now added 4 arms, and shortly, we'll be closing one of the arms. There'll be 3 arms that go on. It'll be ibrutinib, venetoclax, and FCR. So actually, we've now got 3 trials, actually, for the price of 1, or a bit more than 1, because we've not had 3 years waiting to get the results. We haven't got the results of the previous trial. We've accelerated really rapidly. So we're going from the first time we used this combination in 2016 to finishing the phase 3 trial by 2021 or 2020, and then results will come later. So we can really concertina to speed it and move. And what we're planning to do, of course, because it doesn't stop, is continue to do trials, and that's why the Bloodwise is important, the TAP program. And then if we get another interesting one, move over to the next one. And hopefully by this time, chemotherapy's gone, and we're trying to move on to the next combination and beat that, and get more patients with shorter duration of therapy and potentially negative remissions and moving towards cure. So really that acceleration is what we've done in the UK very efficiently, which other groups have struggled to do for a variety of reasons, and that's what's been important. So if you look at the novel therapies, this is the last slide with anything on it, there's lots of treatments coming along that are being used for diseases like CLL, some of which you'll have heard of. Oops, go back one. I won't go back. So, yeah. So, and we're testing these now in phase 2 trials and phase 1 trials, and then, if necessary, moving to combinations quickly to get to cure in time for all the patients to benefit from it, rather than waiting 10 years. And this isn't a one-person show. This is the whole UK working together, and in red are various different the CLL group, including consumer patient representatives and all the big centres in the UK. And then we have the Bloodwise for the phase 1, phase 2. We have Leeds, CSLU here, big team of people doing the phase 3 trials, and then the labs, and then the companies. And so it's really an effort that we're doing together, and it really has accelerated our movement in trials. So that's where we are in CLL.